recording. Okay, so welcome everyone to this population dynamic meeting. Today we have Orkun Soyer as a speaker. I will share my screen to present the speaker. I suppose actually you can let me try to put it full screen. Okay, I hope you can see it full screen, right?
Yep.
Great. So Orkun Soyer from the University of Warwick in UK. The title of the talk is Physics of Cyanobacterial Motility Leads to Macroscopic Formation and Influences Assembly of Spatially Organized Stable Communities. So some few words regarding the career of Orkun. So he had a PhD at the University of Michigan, US, then postdoc in Zurich, TH, and then he moved to Italy in University of Trento as a researcher and lecturer at the University of Exeter. I'm not able to pronounce it properly. Professor at the University of Warwick is actually his current position. And the goal is of his research is to predict and give a quantitative understanding of cellular metabolism and metabolic interaction among cells. So he actually used a combination of experiment and mathematical modeling to study the communities and the cellular metabolism. And among the many, many different projects and topics that he deals with, there are emergency and evolution of metabolic excretion and interaction in microbes, species coexistence and metabolic interaction in spatially organized microbial communities, and mechanism of metabolic interactions among soils, microbes, and plants. And today, so I will chat about one of these topics actually, about the physics of cyanobacterial motility. And I will leave now the stage to Orkun. Thanks again to be there. And if people would like to have, if would like to chat during the question, probably you can actually put your question in the chat or um, just raise your hand. There is a command here, or open the microphone. We will have also discussion in the end. So please, Orkun, the stage is yours.
Right, thanks a lot, Marco, and thanks for the invitation. So as we were just chatting, you guys have been doing a great job with the series. I've been attending to many of the talks. Well done, and thanks for having me. So yeah, I have a handful of a title there. I'll straight go into it and try to explain what I'm on about. And but before I start, Marco has already mentioned quite a few topics. Actually, I don't know if we work on all those, but the way I see it, I mean, we are interested in communities and microbial metabolism or metabolism in general. I'm not going to talk about metabolism today, but I thought I'll just put a slide up and explain a little bit about why we are interested in that. Of course, you know, in the context of communities as well, we know a lot of interactions are about through metabolites, and we are actually at the sort of cellular metabolism side try to understand these metabolic excretions, why they happen, and essentially they go through the changes in metabolic fluxes, and this is something we are quite interested in understanding. But yeah, so I'm happy to talk about those topics if you want to email me or grab me after this talk. But yeah, today's talk is about microbial communities. And in this series, we had many talks about this topic. So I think the audience here are quite familiar with communities. What we've been sort of thinking about in this context is actually the dynamic nature of microbial communities. How do they change over time? How species coexist or not? And how sort of environmental factors influence these interactions. And over the years, I mean, we have sort of published with others as well, sort of opinion pieces and also organized large meetings, residential meetings in this area. So I put these up here in case people want to sort of look up some of these events. And actually, we organized 2 meetings also with Rosalind at the Isaac Newton Institute, of which the talks are still available. If people want to sort of see what we were talking about 5 years, 10 years ago, maybe it is sort of an interesting exercise. So within the context of this dynamical systems view of communities, I think one of the things we got quite interested lately is the influence of space. And where people have looked at spatial aspects, there has been always results that suggest that spatial organization can change and affect a lot of different things. For example, the coexistence of different species, but also sort of their interactions when you have a spatial structure. And one way to think about this is actually, again, linking it to metabolism. When we have microbes excreting these compounds or changing their environments in other ways, for example, through influence of pH or oxygen consumption, those changes will immediately affect the other microbes around them. So actually, in a microbial context, we think of a feedback between cellular physiology and the environment, the microenvironment. And this feedback is quite interesting in my view, and we like to understand this more. I'm not necessarily going to provide an understanding of that in this talk, but that's sort of the direction of travel, if you wish. And I like to sort of mention this talk from Sunil Laxman's group in India, where they actually looked at this a little bit in a yeast colony. So it's not even a sort of a complicated community with many species, but just a monoculture actually grown in a spatial context. And what they discovered is actually cells differentiate phenotypically in that environment and change their metabolism according to where they are in that colony structure. And this, of course, relates to oxygen gradients and so on. But this is a nice illustration of this kind of feedback that I'm talking about. As I said, the talk is not necessarily going to be on those feedbacks, but more generally, how can we understand the influence of space in a sort of a community context, and hopefully also sort of getting closer to naturally existing communities rather than just looking at a single species or few species. And sort of the motivation for this is quite obvious. I mean, everything and anything in nature seems to be spatially organized when it comes to microbes, and these spatial organizations happen at different scales. We have aggregation of within sort of single populations or co-cultures at sort of cellular scale, micro scale, but we also have macro scale formations that are then driven by these microscopic interactions. And I put up here examples. Many of these are from cyanobacteria, interestingly, and I'm going to talk more about cyanobacteria. But I guess that the concept of spatial organization and the questions we're going to ask within that are not just related to cyanobacteria. But curiously, it seems that cyanobacteria do tend to form these kinds of structures, both in freshwater environments and in oceans. So I put up some sort of general questions here. Again, I'm not going to answer all these questions, but this is again the kinds of questions we would like to ask and understand. You know, how do these structures form, and what are the dynamics within those structures? So to study this, actually, we picked up a system that comes from a local reservoir to us. So this is a Draycote reservoir. I show the picture there at the top. This is near our university here. And what is curious about this reservoir is that throughout the year, you have these aggregate structures within the water column. And these structures are visible on the left picture there. Hopefully, you can recognize them. And this is not necessarily like a bloom station, a situation that comes like seasonally or something like that, or occasionally, but rather these structures are present throughout the year in this reservoir. And, we started to look at this, just out of curiosity as a Friday experiment, really, when we first started to look at these structures. And then we got really interested in understanding more, and I started to write grants. And finally, in 2020, we got a significant funding from Gordon and Betty Moore Foundation, which allowed us to really systematically study these structures. And what we have done is simply to bring those structures into the lab and try to grow them in the lab in a sort of a defined media. Try to basically create a sort of a model system. I don't want to claim any ecological relevance here. I see the system more as a lab-adapted toy system, if you wish. And of course, some elements will have some resemblance, some linkage to nature. But these structures form in the lab as well, and we don't think that they are the same structures that we see in the reservoir, but there is definitely the structure formation in a consistent manner as we subculture and regrow these systems. And what we have discovered very early on is actually these structures are not static. So they are able to move. And this video is actually taken over a day, and it's not somebody closing the door of the lab and shaking our cultures or anything like that. This is sort of the directed or sort of non-Brownian motion here. And you can see that there are different things going on. There are some film-like structures that furl up, but there are also granular structures that sort of move about. And whether they're going somewhere or not, we are actually still studying that. But what is fascinating here, at least to me, is that we have microscopic bacteria, and these structures are macroscopic. They're about between a couple of millimeters to centimeters. So they must be harboring, thousands, if not millions of cells. And how does this movement happening? So at this point in this project, we were like, okay, so we need to really understand the physics behind this. And so our original questions about sort of microbial community dynamics and ecology sort of still remaining. We sort of split the project, if you wish, into two halves and wanting to understand the biophysics. And hopefully the two ends will link to each other. And my talk is actually switching between these two topics, and it's broken into three parts. So I'm going to first tell you a little bit about the motility of actually the cyanobacteria that is the dominant species in this system. We know there's only one cyanobacteria in this particular system, and it is a filamentous cyanobacteria. It looks like that under the microscope. And if you take an EM image that is also shown there, you don't see any flagella or pili. So this motility of this filamentous cyanobacteria is so-called gliding motility that people have described in the past. And we know this structure formation, the large-scale structure formation, is linked to this motility. So in the lab, we ended up with another culture that has a mutation, which we identified in this cyanobacteria that makes it non-motile. And that culture does not form these structures. So that's how we know the structures are linked to the motility of the cyanobacteria. So the gliding motility has been studied in different organisms, mostly in actually single-celled bacteria, where people think this actually is achieved through motor proteins that are traveling across the cell surface on a helical track. So this is actually a sort of a working model. We still don't fully understand the molecular basis of gliding motility, I would say. But this model posits that there are traveling motors on this helical track, and when they get caught between the cell and the surface, because they're on a helical track, they translate their own travel into the motion of the cell, and the cell rotates as it moves forward. And the characteristic aspect of gliding motility is that the cells secrete slime, and people think the slime gives an addition between these traveling motors and the surface. And actually, if you mutate slime production, either through biosynthesis or excretion, you lose motility. So this is most of this information comes from Myxococcus and Flowobacterium, which are single-celled organisms. And of course, here in the case of filamentous cyanobacteria, we have multiple cells. So the situation is possibly more complicated and different, possibly. And again, the molecular mechanisms are not very well understood. So some people think that the gliding motility in filamentous cyanobacteria works through pili extrusion and extraction. Some people think that it is actually directed the slime excretion that pushes the cells forward. We don't think that that actually physically works, but the pili-based mechanism can be possibly at work. The cells definitely have the pili apparatus, at least the excretion aspects of it. But again, things are a bit unclear. And of course, in our case, we have a sort of a non-model organism, something that we have found in nature. So we don't have genetic capability, so we can't knock proteins off and things like that. So we thought at least we can maybe look at the dynamics of the motility, because many of the studies actually have not necessarily looked at trying to characterize gliding motility itself. So to this end, we started doing, extensive microscopy, and we can look at the cells, filaments, I should say, moving on agar, and on agar they move back and forth in a very sort of a defined manner. And on glass, this movement is a bit more free, and they are not confined on a single track. But again, you can see a back and forth movement most of the time. So the cells tend to go in one direction and then reverse and go in another direction. And we can quantify this by just tracking the filaments. And then what we see is that actually you're looking at the position of the cell and the distance traveled and the speed. As I said, the cells go one way and then stop and reverse and go another way. We can quantify this stoppage time, which is shown in gray on the lower graph. And we can look at the distribution of that time. And it seems that most of the times, actually, the stoppage is quite short, but sometimes filaments spend a long time being stopped before they reverse, go the other way. And the speed of the filaments doesn't seem to depend on their length. So based on these observations and a few other observations, we thought maybe we can create a model and explore some ideas. And we collaborated with Marco, Chantal, and Emmanuel, three biophysicists, to create a very simple sort of a bead and spring model. And I'm not going to explain this because I don't have time, but essentially two key parameters in this model is that we assume that cells basically generate, each of the cells in the filament generate a force. This is motivated by the observation that the speed and the filament length don't correlate. And then the second aspect is that cells are mechanically coupled, and the forces between the cells will be sensed and cause a change in the direction of the force that each cell generates. So there's a mechanical coupling assumption, and there's an assumption of each cell generating an individual force. In addition, we introduce a sort of a memory parameter in the model that sort of controls how quickly cells will respond each other's force change in direction of force. So we can play with those two parameters and see if we can generate basically dynamics like we observe in the experiments, especially this sort of reversal frequency as well as the time spent during reversal as in the stopped form. And actually the outcomes of this model is, as you might expect if you're sort of physically minded, the coupling strength, if you increase that, you get a certain response, and if you decrease it a lot, and if you also reduce the memory, you get very erratic movement. So these are similar plots like I previously showed, but maybe a movie is easier to grasp. Basically, based on these two parameters, this model can get stuck or generate actually reversals just like the original filaments. But one thing that we learned from this model is that this coupling, there should be a good amount of coupling between the cells. And that's something, and also this memory parameter is important. We don't have ways to experimentally probe the memory parameter, but the coupling parameter, we thought we can try to experimentally probe. So what we tried to do is use turf microscopy, which gives you a very small illumination such that you can look at membranes of cells. And we thought we can apply this to see if we can actually track individual cells. And to our luck, I will say, we found fluorescent membrane complexes on the surfaces of these proteins. And these allow us to track individual cells. And when we did so, and combined with the way the turf works, we actually discovered these cells are rotating, just like people have observed with Myxococcus and Flowobacterium as they travel. So the rotation and translation is fully coupled, but also now we can use these fluorescent complexes to track individual cells. And when we did this, we found that the cells were actually quite coordinated in their motion through these. And actually here we are seeing a reversal event in this movie. But the turf microscopy is limited in the sense of how much of the filament we can look at. And if we could sort of extend this, perhaps we thought we could see some decorrelations among the different cells. This was motivated by the sort of finding that actually, if you look at far apart cells, there was sometimes a delay between their movement, but it was hard to say if this was noise or a real phenomenon. So we tried to go to longer filaments to ask this question: are the cells always coordinated? And when you look at really long cells, actually, we found that sometimes these cells can buckle in the middle, and we can track the two ends of this long filament. It's hard to see in this movie, but if you analyze the data, you actually find that at some point in that movie, the two ends of the filament are actually going against each other. So they're completely decoordinated, and that's where this buckling happens, and actually coupled with the rotation of the filament, this ends up forming a so-called plectony. So it twists on itself. Now, this twisting is interesting, and we observed it on many occasions when we started to look at longer filaments, and especially on glass, you tend to get quite a few of these plectony formations and buckling events. And this is just one example of that. And when that happens, the movement of the filament can get quite erratic. So I'm going to come back to this point, but now I want to switch gears and talk about the microbes a little bit. This is, as I said, a sort of a natural system that we picked up. And as I was explaining, we brought this system into the lab. And what we've been doing in the lab is to actually subculture this system over many passages. So it's, if you wish, a bit like a Rich-Lansky experiment, experimental evolution experiment, where we just sort of passage things over time. And we used quite a long-term culturing. So each passage is about 30 to 40 days, but sometimes we used even longer time spans. So we really were trying to sort of keep the system and, you know, don't select for fast growers or things like that. We also used a defined media that lacked carbon. So we deliberately wanted to have interactions among different species in that way. This was done over almost 2 years, and we have quite a bit of data from this system, but I'm going to just explain part of it. So when we looked at the sequencing and look at the coverages of the different bacteria, so we are quite certain that we are actually seeing the full diversity in this system, because when we bin the sequences and create genomes, and when we then map the reads from samples onto those bins, we get above 90% recovery of those reads. So we think that we have recovered all the genomes in the system, and there are 17 genomes that we can find. And what is interesting is that those species seem to be coexisting stably over the time that we've been passaging. So, and these are the sort of different phyla that we have. And of course, the main question is now, you know, how do these guys coexist? And they're coming from quite a different parts of the taxonomically, they're quite diverse. The first thing we did to try to understand how they coexist is to look at their genomes. And some of the common things that we found is that many of the alpha proteobacteria had anoxygenic photosystems, which is not something we were expecting. And many of the bacteria had carbohydrate degradation pathways. And then we also looked at vitamin biosynthesis, which was variable. And also we found some sulfur oxidation genes. Now, the sulfur oxidation, some of the sulfur oxidation genes work in anoxygenic conditions, and also the anoxygenic photosystems are usually require anoxygenic environments. So we were then curious about the oxygen in these structures that we are seeing. And what we found is that many of those structures, actually, when you take a probe through them, a microprobe, you find their interiors are actually oxygen-free. And we could also sort of image the bacteria to some extent, and found that most of the times the exterior is occupied by the cyanobacteria and the interior is occupied by bacteria. So this suggests that actually there could be anoxygenic niches within the system, which could be perhaps allowing some of the bacteria to coexist with the others. And to see if we can perturb this, we started cultures that were shaken, replicate cultures that started from the same ancestor and then kept either under static conditions, which is our normal condition, or under shaking conditions. And we saw some changes in the coverages, and we're still trying to analyze this. The other point we were curious about is about these degradation enzymes. I didn't talk about this in the motility context, but the motility, a little bit, I said already, the slime. So the slime is a requirement for gliding motility, and this filamentous cyanobacteria also secretes slime. We can see it a little bit on phase images, but we were also able to strain this and find that actually filaments are sort of inside these slime tubes. And what we then found is that many bacteria, I don't know how many, but bacteria, at least some of them, can actually get attached to these slime tracks. And we can visualize this to some extent. We also then tried to extract slime and analyze its composition. We found that it contains galactose and glucose, and galactose was actually the main monomer in it. And several of the isolates that we could isolate from this system could then grow on galactose. And then finally, these degradation enzymes, for many of the bacteria, they were actually targeting galactose. There were other degradation enzymes as well, but so we can't sort of conclude a specificity here, but at least it seems that there is the possibility of breaking down that slime and utilizing it as a carbon source. And as you would expect from this, that if we then add glucose into the system and again create this kind of experiment, perturbation experiment, we did find changes in the composition. And actually, I'm showing here the third subculture, but we took this experiment to the fifth subculture, and you almost completely get basically a wiping out of the composition. The cyanobacteria really diminishes, and all the other bacteria come up. So that suggests that this stable coexistence is partly achieved by structure formation and partly achieved by the lack of carbon. I also mentioned vitamin exchanges, or vitamin biosynthesis pathways, and what we have seen there is actually only one of the species in the system has the capacity to produce almost all of the vitamins. And for other species, the biosynthesis capacity is variable. And now, based on this, actually, there are two species here that have possibly complementary biosynthesis capacities, and we happen to be able to isolate those. So we could actually create an experiment where we can show that one of these species, the Pseudomonas species, can actually complement the auxotrophy of another species in terms of vitamin B7. So based on these findings, we are proposing a sort of a working model of this system, and it remains to be seen how generalizable this is. But I think one possibly common feature is this oxygen gradient that emerges in these systems. Even though cyanobacteria is providing oxygen through photosynthesis, there is a very strong oxygen gradient forming across those structures. And this could create niches for the different bacteria. And then on top of this, you can have these different metabolic interactions. And of course, there's also a light shading effect, which people have looked at, for example, in cyanobacterial mats. The red light can penetrate deeper into these structures. And that's actually the light that usually is used by anoxygenic photosynthesizers. So I want to come back to sort of the motility aspect and think a little bit about how these aggregates form in the first place, because we think that the aggregation and the structure formation is an important part of the diversity here. We like to understand that a little bit better. And to do so, the challenge is we need to look at the motility of the cyanobacteria, but we need to look at this process that is happening at a macroscopic level. So the next movie I'm going to show you is already displaying this problem, that we have to create basically stitched larger fields of view to be able to see both micro and macro processes at the same time. And this next movie going to jitter a lot because it is a sort of a stitching of many many fields of views, and the stitching didn't quite work well, but it's a nice video, so that's why I wanted to show it. And in it, we can see almost individual filaments, and we can slightly see these plankton formations that I was mentioning earlier. And it seems that through those kinds of plankton formations, filaments can get entangled, and they tend to form these central sort of aggregations, which are then connected to each other through other filaments. So you create a system where actually different pulling and pushing forces can interact. And we wanted to, again, understand this a bit further at a larger scale. So to that end, we started to film culture flasks rather than doing microscopy. And we wanted to particularly understand how these larger structures form. So this is a movie where we take pictures of a beaker from the top, and then I'm just showing a cutout here, taking out the environment and just looking at the bottom of that beaker. And this is a sort of a culture after subculturing. So we just inoculated a bit of cyanobacteria. In this particular case, we also had glass beads in the media, just to be able to sort of see if there are any liquid flows happening or anything like this. So try to ignore them. They're not important for this video. But what is happening here is, so you can see some of those glass beads in the surface floating, but we are actually looking at the bottom of the glass beaker, and you can see some of the cyano aggregates already, large green aggregates. They are sort of moving about, but also hopefully you can see some yellow particulates emerging, and some of those yellow particulates are moving as well. So if I zoom in into one part of this picture, what you realize is that actually the green aggregates of cyanobacteria, they seem to be actively pulling those yellow particulates to themselves and incorporating them into their aggregation. So this was quite fascinating to us. We first wanted to understand what those yellow granules are, and then secondly, we wanted to understand again, how it can physically work that these aggregates of cyano can bring these particles to themselves. So I'm going to tell you already that the yellow particles are iron minerals. They get precipitated in the media even without bacteria, but they are never collected in this manner when there is no bacteria. So we are now trying to understand how this works at sort of a micro scale, and we found two different mechanisms, one of which is actually, again, involving the slime. So filaments can drag particles that attach to the slime, and we can visualize this to some extent by staining the slime. On the right picture here, you see some beads, and then the green staining is the slime. And what we are seeing in these pictures are also the iron precipitates that are tiny compared to what we were seeing earlier. So these are early stages of these cultures. The second modality of collection we found is relating again to the plankton information. So I'm going to play this movie, and we got here, this is false colored movie. The white particulates are iron minerals. The black ones are beads that we put in, and you can see filaments as white as well. And this filament in the middle of this movie is getting stuck on those particulates, and also then starting to form planktonines and bucklings. And as it does so, it's a bit like a breakdance. It ends up collecting a lot of particles, basically. So we are trying to, at the moment, quantify these processes and see also if we can model them somehow. But what it seems to be happening is that through this motility, and specifically the physics of that motility, these filaments end up collecting particles from the environment and incorporating them into the aggregates. So I already mentioned the minerals are iron, and this is quite interesting because iron is relatively rare in the environment, in most environments, and it is generally suggested that cyanobacteria especially can be starved for iron because they do have a high iron need for relating to photosynthesis and other processes. So to this end, it's interesting to see that they are able to collect actually iron minerals. And we then started to check sort of the biological significance of this. One curious thing about this cyanobacteria is it lacks iron 2 transport. So the iron provided in the media is iron 3. Usually iron 3 is insoluble and bugs need iron 2. So they will either reduce it outside and then take it up, or take it up as iron 3 and then reduce it. And one way of taking iron up is through sderophores, which can bind iron 3 and actually grab it from its insoluble state. And curiously, this our cyanobacteria has sderophore transport genes, but doesn't have sderophore genes. But the other bacteria in the system, some of the 17 that I mentioned, do have sderophore biosynthesis clusters, and they also seem to be expressing sderophores, which we can quantify with this assay, but we are also further trying to quantify it better using mass spec. So to wrap all of that up is basically what we are thinking in this particular case is that the motility of the cyanobacteria results in the formation of these larger structures, and which generate both environmental niches, microenvironmental niches, but also an opportunity to interact, especially through the slime or through other means. And we end up with a specific community forming around that cyanobacteria within these structures. So we think that this could be a general phenomena. And if that is so, then we think that you should be able to find this dynamic over and over if you were to rerun these types of experiments. So then we thought, well, we can actually try to do that. So we went back to the lake, and actually we also run a small project of sampling the lake on a weekly basis, just to also look at the microbial ecology in the lake. But as we did so, we also picked up cultures or samples and started cultures in the lab just to rerun the whole experiment again. And actually, we are doing this now under different conditions as well. So these experiments, again, have been going over 2 years, and we are just starting to analyze them and see if we get a consistency in the composition of the community, but also in terms of the functions of the community. In many of these cultures, actually, filamentous cyanobacteria do well, and we do get structures to start with. So I think that's where I'm going to summarize things. I started with sort of these high-level questions. I don't have answers to all of them, of course, but we think motile filamentous cyanobacteria are interesting in terms of generating these structures. We by now know there are other cyanobacteria in the lake that could also generate structures, coccoid cyanobacteria. We're trying to go through that data as well. But at least under the conditions of the lab, these motile filamentous cyanobacteria can definitely generate structures. And those structures definitely have a bearing on the communities that form. And I think one of the biggest take-home messages is that these, so we call them enrichment cultures, as microbiologists will do, can act as model systems, given that they give us really stable sort of mid-complexity communities. And we can ask many questions about, of course, population dynamics and evolutionary processes as well. I find the whole thing interesting as well in terms of symbiosis or sort of strong interactions. The Gordon and Betty Moore funding is actually hinged on the emergence of symbiosis. I think this kind of structure formation can stabilize the environment perhaps and lead to emergence of sort of stronger interactions in the long run. So it could be the beginnings of some interactions that then get stabilized. All right, so I'm going to stop there to take questions, but I like to put this ad up here. Some of my colleagues are actually organizing the first conference on cyanobacterial granules. They call them photogranules, and it has a bit of a biotechnology spin, but some of them are microbial ecologists. So if people are interested, please check that out. It's going to be at Delft in Netherlands. And with that, I'll stop and thank you for your attention.
Thank you, Orkun, for the very nice talk. So I will